Let's talk about variation in the gametes. So this is going to occur in meiosis, right? So this is really important because as humans, we need to understand this because we're large multicellular eukaryotes that live a really long time. So that means that we don't get to take the advantage of multiple generations in a short amount of time, being able to evolve and adapt to our new ecosystem. So what we have to do is we have to employ sex. So sexual reproduction allows us to mix genomes from two separate individuals who have been tested by the environment, and it allows us to arrange the deck, right? So there's randomization that also plays a little bit of a role in this. So it's that randomization that we're going to look at primarily today. All right, so you remember meiosis. So I want you to take a quick look at meiosis 2. The first thing I kind of want you to remember actually is oddly from meiosis 1, where we had crossover events, right? So we had crossover. So these points exchanged and these points exchanged over here. And then this one exchanged over here, right? So crossover in meiosis 1, right? So it's a prophase, prometaphase type thing, right? For the most part. So it's occurring generally the lion's share is occurring during that phase. So for the rest of it, what we're doing is we're starting to look at how we're going to just randomly chuck different genomes into the forming haploid cells, right? So here's little, sperm or egg cells. Okay, so here we have everybody aligning on the metaphase plate. So notice the first thing that happened was after we've separated this cell, we have different chromosomes going into different lineages. As these chromosomes align on the plate, you'll notice we still have, sets for each one. So here's like maybe chromosome 1. All right, and then maybe here's chromosome 2, and here's chromosome 2 over here, and then chromosome 3, and then chromosome 3 over here. Right, so see how they're we have that split. So now metaphase is going to lead us to anaphase or backphase, and we're actually going to split them again down the middle. So now just take a look at this. Okay, here we go. Put my highlighter on. Okay, so let's look from chromosome 1. Okay, this highlighted area, this highlighted area, this highlighted area, and this highlighted area. None of these are the same. So they're all very different. They're all holding, here, let's get a pen, actually, that'd be better. Okay. They're all holding different genetic information, right? So here's like the first two. Okay, here's this one, right here. This one is right here. And then the next ones, let's see here. Ooh, right, this one. And then this third one, none of these look exactly alike the others. Some have entirely red bodies, some entirely blue. Some, in this case, are going to have a blue body with red tips. The other that also participated in crossover is going to have a red body with blue tips. See how that's different? Oops, that was supposed to be blue. There it is. Okay, so there's that level of variation. And then the last one, right, is when we complete this process. So this process is called independent assortment. And what's important to know about independent assortment is it's random. So now we have 4 distinct, genomically distinct gametes that can be used to produce 4 independent individuals. So that's one of the important ways that we get variation in the gametes. It leads to some of these different possibilities, right? So possibility A, we have blue all on one side. Possibility B, blue's on, say, 2 different sides, right? Also, red, one side, red mixed on 2 different sides. Just simply that arrangement can cause this new possibility for being able to generate new individuals. Let's watch them separate out. So look at these different combinations, right? So here's combination 1, combination 2, combination 3, and combination 4. And this is not even accounting for crossover, right? This is just the randomization, the random independent assortment. Now let's look at the power of crossover a little bit in conjunction with independent assortment. The first thing that's happening is we're going to divide, right, our duplicated chromosomes, and we're going to imagine these as going into different haploid cells, right? So imagine these were going to different sperm or egg cells, right? Okay. So genetic recombination, right here, is going to allow us to recombine genes from different parents in the process known as crossover, right? So that allows us to get the different genes from mom, from dad, and kind of mix up that deck a little bit more. So one of the places that's important is that central location where we actually have this crossover point. It's called the chiasma. If you have more than one, it's a chiasmata, all right? And that's the site of exchange. So here you can actually see a photograph. Looks pretty good, right? You can see, here's from a karyotype, right? So it's chiasmata, right? Two of them, chiasma, singular. And as you can see, this is where they're actually exchanging their ends. So now let's look at what the variation means in terms of independent assortment and recombination. So here we have mom and dad's chromosomes separate. We're going to have a breakpoint. We're going to form the chiasma. As crossover is completed at the chiasma, we now are going to have these like different recombined chromosomes. Variation city, right? That's great for us. So it means this. Now we're going to not only have these different arrangements going into different gametes, but they're already going to be recombined too. So now that's what makes them completely individual. All right, so I hope that's helpful in understanding how that variation is exploited in a couple of points during meiosis. And great, if you have any questions, please feel free to email me.